has probably been like one of the most important things I've done in my life.
And you're only how old?
14, almost 15.
Right. So people who are 14 and 15 are saying that they feel that it's one of the most important things they've done in their life.
Yeah. This plant is eelgrass, and plants like eelgrass only inhabit 2% of the ocean, yet they're responsible for 50% of carbon sequestration. And that's when plants take carbon out of the water and atmosphere and bury it back into the earth, slowly stopping the effects of climate change. 1 acre of eelgrass can absorb as much carbon as 40 acres of a terrestrial rainforest. This plant lives on 4 of the continents in the coastal ecosystem. Eelgrass creates a necessary habitat for hundreds of animals, and eelgrass beds also reduce the effects of coastal erosion.
So we're going to latitude 42.247123, longitude negative 70.780309. We're going to investigate some eelgrass and do a test on whether our new technology, being Canway pingers, etc., is functionally working for measuring eelgrass, pinging it, etc.
So let us turn on the Canway pingers and we'll see at least whether they will make a proper track. And I would like to see if we can do them in order.
These are Canway pingers. They, whilst we turn them on, they communicate with satellites to record our location, as it were. You turn them on here, and you can engage in making individual points by which correspond to, in our case, eelgrass in every moment we're over eelgrass by pinging it here.
The other thing that's funny about GPS things is GPSs don't do so well in the fog, believe it or not. So this is basically what low tide in Cohasset Channel looks like, and this is a minus 0.2 low tide today at 6:19, and oh, it is 6:21. Let's get the GoPro ready on the quadrat, and we can pass it in to
What's the time?
7:52.
Okay, 7:52. Let's look a good place to
No,
time in.
6:52. I'm sorry.
Oh.
6:52.
6:52. And this is going in. What's this giant patch in the middle?
That's a great question. So this patch, it looks all poofy.
Yeah?
We've been calling this red filamentous algae, and this is exactly what it seems to do. It seems to gather, it rolls around like tumbleweeds, and it seems to gather in sort of a basin.
Can we try to collect a sample?
Sure you can. And we're wondering what its impact is on eelgrass and other things, because it seems to block out a lot of the light when it's really thick like that.
Like harpooning a whale back in the old days.
Tell me when you have it.
Have it? Oh wait, no. This is the worst way of doing it.
Oh dear.
Alright, we're gonna head home. But this is a good corner to be in because we're pretty close to the very low tide line. This is an area of question. That right there, those two brown rocks are the twins, and the next big rock is Hog Rock. There's sort of a mystique about science from the days of yore, that it would be people in white lab coats that walk around sounding like Dr. Bunsen von Honeydew. And the reality is that science is much more open than that. That you can be somebody who's very detail oriented. You can be somebody who has curious questions that drive scientific inquiry. You can be somebody who communicates science to non-scientists, which is a really valuable role to play in the world. So people come to CSCR with filmmaking skills, they come to CSCR with curiosity, they come to CSCR with an interest in being outdoors, they come to CSCR with dyslexia, and they want to be able to do something where they're not feeling like they can't do it because of that. So the different roles that we create, or they create on a research team, allows people to participate in many different ways.
For the expeditions in CSCR, they vary quite a bit, but we try to use some consistent tools that collect consistent data. The first one we use is an app called Survey 123, which has a multitude of questions to help us capture information about eelgrass and then places it on a GPS coordinate, so then we can show it on a map. It's probably the most useful tool we have because it allows us to present our data. Another tool we use regularly is this, what's the name?
Lead line.
Yeah, this is the lead line, which we use to measure depth, and in previous years it was used to find the bottom type by filling this cavity at the bottom with cream cheese, and then it was thrown into the ocean to pick up the debris at the bottom, showing the bottom type. One of the most important tools we have are these quadrats, which capture a consistent image of the eelgrass in a square. This allows us to accurately measure it and say the percentage of density in the eelgrass bed. Another tool we use is the hose stick. We use it to look into the ocean with a GoPro attached to the end. It allows us to look under the waves if a human can't swim, and allows us to record footage, which we can analyze back at the base. Okay, I'll swim the tide line.
Nope, you're gonna walk the tide line because you can't swim it.
Well, can I swim next to the tide line?
No, you have a job to do. We're gonna mark the tide line
Alright.
by going 1 foot in and 1 foot out.
Sure, you know I'm gonna go one way, you go the other way, or
So,
I'm gonna go a different way?
We're gonna go the same way.
Okay.
Because Nikolai and I already did this one yesterday. So, so here's your Kenway finger, and you don't need to actually push any buttons in it because this will do your track.
Yeah.
Okay. So, COM 10 is going to Max. COM 11 will go to Owen.
Am I talking?
You can, yeah. Just don't lose it.
Well, I noticed that most of, if not all of the rocks had a fair number of tunicates on them, and so I'm wondering how that would impact the number of tunicates found on the eelgrass.
I didn't really find much, but I did notice that all the eelgrass had a lot of calciferous coverage, which made it look really gray, and a lot of the rocks had some sort of coverage too, some sort of bumps, which I couldn't really see, but I could feel on my feet.
So I noticed that there was a lot of small bits of seaweed growing right at the tide line. Some, which we think are sugar kelp. Sorry, I'm distracted because I'm trying to drive without damaging the eelgrass. And toward the twins, there's a big area of sand. I saw some blue mussels, a good collection growing on the outside of the twins, and I saw some European oysters, but no Native American oysters. The year that I joined, Laura Humphrey, who teaches a very popular statistics class at Cohasset High School, had students walking across hand in hand out into over the flats in the shallow water until they got to eelgrass. And then at that point, we were using like exercise apps on phones, and not all of us even had smartphones at that point, but we had an app called Map My Run. And we didn't know maybe exactly how it all worked, but we knew we could make a little map if you followed a route. So we were trying to use that to outline the eelgrass.
Well, we wanted something to do with the sea perch robots. And since my background was in science and in math, I wanted something that we could collect data about and then analyze the data. And it seemed like the best idea because we could get there close to shore. We didn't have a boat with a motor at a time. We just had a few donated kayaks and paddle boards, and I used to bring my 2 paddle boards. And we would just load, we'd swim across to the island there, then we'd drag those boats down, and then just a few people would go out and snorkel stuff, and we'd throw our square to get a random sample, we'd cut all the eelgrass, and then we'd count the number of tunicates.
We're thinking we'd go out and do the shallow edge again, which is what we did yesterday, and we'll do 2 teams going along the shallow edge. And one decision to make is, do we want both teams to be following each other, so the second team is kind of fact checking the first team, and we can see if we all agree on things, or should we go split 2 directions and then get more data? I think we said before that we should have 2 teams doing the same thing, so we see if it really works.
Yeah.
All right, so Max and Nikolai, are you guys ready to swim?
Yeah.
Okay. And Max, can you explain what you did yesterday about that?
I already did.
Oh, you did? Okay. Yeah.
Wait, let's go.
Oh yeah, turn on light. This is smells like hell. It's so white. Yeah,
We need to bring the dinghy around. It's actually easier to have it in the dinghy. Is anybody feeling confident about rowing the dinghy, or you want me to do it?
I'm not confident. I am a single.
Can you describe to Nikolai a little better what they need to do?
So Nikolai, your job will be
To swim.
to
You
swim.
need to go.
So we're gonna give you a cannaway pinger, and you're gonna need to follow exactly along the edge of the shoreline edge of the eelgrass. To determine. So you're gonna stick with the main patch and not follow any outliers. That's the people in the canoes job to ping them with survey 123. So what you're gonna need to do is follow along. If something is more than 2 feet apart from the next closest chute, then we're gonna count that as an outlier. So just kind of going a straight line. It's going to be a little difficult to get acquainted. So we're going to go almost all the way into shore, then come out. So we're going to make sure we have the farthest out shoreline edge of the eelgrass to track.
All right.
That's it.
And always face in the same direction so you don't get turned around. It's very hard. It's very easy to just get turned around to the eelgrass. It's just this. You have no bearing of direction.
We're en position.
Hopefully my neck won't be hitting you.
no, sit down, sit down.
I'm sitting down. Hey, which shot's the fun? Which one's the We already do. We have.
And if that camera thing doesn't seem to be working,
Yes.
yeah, but right, so do we? And then you guys have the CSR phone, right, Noah?
Yeah.
Okay. And then we're going to
I need to get ready for my matter work.
So we're back. accuracy of the GPS may be even less if it's underwater. We'll have to figure out that when we get back. But we haven't made too much distance from our starting point. So we've been at it for an hour. Okay, so we're going to turn those off now so we know that their job is done. Get those. Alright, so let's head back to the boat.
During the last expedition, the CSCR team tried to track the shallow edge of eelgrass beds. That's to say, they used GPS trackers and walked along the edge of the eelgrass beds, keeping the shore to one side of them, and the edge of the beds on the other. Unfortunately, this did not go as planned. For one, the GPS trackers did not work well underwater, leading to the data being skewed. The beds were also difficult to navigate, with small passageways that could look like an edge, but would lead you into the eelgrass bed. Although this didn't work as intended, we did learn valuable information about the layout of eelgrass beds, and we are eager to learn from our mistakes.
We're going to it.
Okay, and then how many is that? What are we in feet, miles?
Miles 2014. We are going to a site that had 50% eelgrass on certain plants to analyze if there are still more dead eelgrass there from wasting disease. Previously, Mr. Buckley has found heaps of dead wasting disease on the floor of there. Unfortunately, there is rough conditions and cold weather, so we won't be able to swim there. So we will try to employ the use of a GoPro to find what we are looking for. Yeah, we're 155 feet.
155 feet.
But it is the 3rd of August, 2021. It is 3:01, and that is good. It is a bit of a low tide, and we are going to site 16 from 2020. And we're trying to see if wasting disease has wiped out this colony, or if it has even just has been more wasting disease. So have fun out there.
Over the past few months, we've been collecting and analyzing our data that we've collected over this summer. Through the past couple months that this documentary has been filmed, I personally believe that I've learned a lot. I have changed, and I've been enveloped in my eelgrass research, learning everything there is to know about eelgrass and seagrass wasting disease. I've presented our research to various organizations who look over the Quasit and Scituate Harbor. Currently, we're on our last expedition to interview Juliet Simpson, one of MIT Sea Grant's researchers who has researched eelgrass and carbon extensively. But we're excited to look forward to the new year as well, as we have a bigger and larger research team and many plans for the next documentary.
All right, welcome to MIT Sea Grant. Let's take a walk.
Thank you.
We have a few, just to give you an idea of some of the different projects that go on here, we've got a big robotics team that has been working on underwater autonomous vehicles for several decades now. This is one of our older ones that's now been decommissioned, as you can see, but it could go down to about 4,000 meters. So this is all the administrative parts here.
Ah. Wow.
This is the electronics lab where the AUVs are built and developed. So this is one of our larger ones right now. It's a sort of a floating catamaran that ultimately is going to have a drone that goes with it. In this room over here, here we have an electronics teaching lab where we have students and student groups and classes come in, and as you can see, it's sort of kitted out with electronics so that they can build some things. We have here a tank. We call it the square tank, even though it's not square. And we can test vehicles in there, smaller ones especially. And then we have in here a tow tank. This is the biology section of the group. We call this our mud room. This is where we bring in dirty stuff when we come in from the field. And we keep most of our field equipment. Let's see what's fun back here. We have these samples which are in the process of being analyzed right now. I don't know if you can see the creatures on there. We put these PVC plates in the water and leave them for certain periods of time and see what colonizes them, and see if you put it in May, and if you put it in September, and if you put it in February, do different animals colonize? Right, here's the classic quadrat, which we use quite a lot for studying eelgrass and other things. So all that field stuff is kept in here. All our cameras, and this is this is the fun thing to me, is that when you do ecology, there are rarely tools that are made exactly for what you need. So like these are the perfect tool for slicing eelgrass cores, and they come from a kitchen supply store. They're for scraping grills, but they're exactly the right thing also for slicing eelgrass sediments. This is the clean side of the biology lab. This is the room where we have a muffle furnace. This goes up to about 1,000 degrees Celsius. We can't do that many samples at a time. This whole thing is just this tiny little thing because it has to be really well insulated. So depending on what kind of vessel you've got, you can only fit 9, 10, sometimes it can fit in 12 samples in here at a time. It's a bit of a mess right now because we're starting up a new project. I have tried in the past to grow eelgrass in here.
Could you tell us about that?
I would love to tell you about it. I was growing it in a bunch of replicate tanks, and I had a bunch of different light systems. Here are the lights. Tunable LED lights, so we were investigating different, if you have more red, if you have more blue, does it promote more growth of the eelgrass? And what I concluded in the end, because I couldn't get the eelgrass to grow effectively in here, it's just too warm.
Too what?
Too warm.
Too warm.
So I was able to give them the nutrients they needed. I was able to give them salt water. Although we don't have running salt water in here, we can create salt water using something like Instant Ocean or other just commercially available aquarium salt products. And the light seemed to be sufficient, but it's warm in here and they didn't grow very well. So our next step, what we're going to do this summer, is get a couple of chillers and try and set up some circulating systems where we can grow eelgrass and actually do some manipulations in different tanks. We found a bunch of amphipod. Oh, here's a good one. With these polychaete worms. We have not identified the species yet, but these worms really like sitting in the emerald tutu. There's an amphipod too, and that one, kind of a big one. Let's see if there are any other good ones. Oh, here's some more. Oh, here's a mouth. There you go. That's one of the polychaetes. For some reason, this particular species, whatever it is, loves these floating wetlands. So if we put out a network of these floating wetlands in Boston Harbor, we'll definitely have a benefit for this polychaete worm, whatever it is.
Only question is that a benefit or a loss? Because you don't know its effect on other environments.
It's a good question.
Yeah.
Since we don't know yet what this guy is or what its role is in the ecosystem, more of it could, be good for some things and not good for others.
Yeah.
So that is kind of a very brief overview of some of the work that we do here.
Hello, audience. It's been quite a year, I'd have to say. For CSER, we've presented to a lot of people, and we've made a lot of progress. We have a bunch of new people who are excited for next year as well, and we hope to carry on our amazing research. I'd like to thank you for watching this. I love that you've taken interest in this. I'd like your takeaway from this film to be that you are interested in what we do, that you are excited for us and excited for the future of eelgrass. It's an important topic that I feel like you should know about. That's why we've done this whole thing. The reason we do this is so you can learn. I hope you've enjoyed the film, and we'll hopefully be back next year.